So the title of my talk is Targeting Safe Cellular Rejuvenation. So there's a very powerful cellular rejuvenation paradigm that's been touched upon earlier, but it has a neoplastic risk or cancer risk. And there's, increasingly there seems to be a scope to remove this risk, perhaps partially, but perhaps entirely, which is the excitement. So I'll talk to you a little bit about how we're going about doing that. And just to start with this, this paradigm, so basically it's called cellular reprogramming or reprogramming with Yamanaka factors. So Shinya Yamanaka found these factors basically with respect to pluripotency. So he found that a combination of OS, K, and M could turn an adult differentiated cell into a pluripotent stem cell. And he actually, he put up this data that just sat there for about 8 years. And then Samir Chandra at University of Edinburgh came along and used a new technology called the epigenetic aging clock to show that when you express these reprogramming factors, you can rejuvenate from age 60 or biological age 60 down to age 0 in just 17 days. So it takes 60 years to build up that aging in the system, just 17 days to jettison that aging out of the system, and up until 15 days, the cells still retain a degree of identity. So there isn't really anything else like this as far as rate of rejuvenation is concerned. So a really powerful paradigm. Later on, David Sinclair showed that these factors, they didn't just affect an aging biomarker; they could do dramatic functional things. So he showed in an optic nerve crush assay that Yamanaka factors could restore a remarkable ability only available to newborn mice. So if you crush the optic nerve of a newborn mouse, the optic nerve can regenerate, but in an adult mouse, the regeneration no longer happens. However, if you express the Yamanaka factors in that adult mouse and crush the optic nerve, it can regenerate just like a newborn mouse. So you're taking an adult mouse and basically restoring functions that didn't previously have access to. Well, no, it did have access to, sorry. And then in 2016, Jean-Carlos Belmonte showed that transiently expressing these factors in progeroid mice could increase their lifespan substantially. So it was a Goldilocks effect. So too little of the factors, no effect on lifespan. Too much of the factors, and you got these scary-looking pluripotent teratomas. So these are cancers where you got the teeth growing out and hair and all sorts of things. But if you've got the amount just right, you got this nice increase in lifespan. And since this study, there's been some really interesting inroads. First of all, it's been shown that there are alternative paradigms for rejuvenation or reprogramming paradigms. So Jacob Kimmel from Calico earlier showed that a reduced set of factors can have the same rejuvenative effect with less effect on cell identity. He also showed that the warning side of this, which is when you express even transiently these factors, you do get pluripotent cells when you analyze at a single cell level. But there's also some really promising results from Vadim Gladshev's lab, where he's basically found a natural rejuvenation event in early mouse embryogenesis. So the fact there's a natural rejuvenation event, we don't know exactly the overlap with Yamanaka reprogramming or those pathways, but the fact there's a natural event suggests this is relatively safe. There may be reasons why early embryogenesis is more permissive for a neoplastic phenomenon, but the fact that your cells will activate this naturally is sort of a cause for optimism. And just to talk about what we're doing, I wasn't originally involved in cellular reprogramming or aging clocks. By training, I'm a mitochondrial biologist, and I got interested in mitochondria because of Aubrey. So I'd read his book. He talks about mitochondrial DNA mutations, and I bought into that. I was convinced that if we got rid of these mutations, that would be a fantastic therapeutic lever for slowing aging and rejuvenation. In 2017, we set up Shift Bioscience basically to pursue that hypothesis. But something very early on, we wanted to show that this was the best lever. Out of all the different approaches, this was the best lever. And it was actually my investor, Jonathan Milner, that put me sort of put me in touch with the epigenetic aging clock. You know, I wasn't aware of the clock. You know, mitochondria was my world. I was perhaps a little bit too blinkered. And the moment, you know, we discovered this clock, it presented a fantastic opportunity, which was to audit our hypothesis. So, you know, now we could ask the question: if we turn up mutations to the max, what does the clock do? If we affect mutations, does it affect the clock? And so we found the nearest lab, and it happened to be the only lab that had a mouse epigenetic aging clock. So Wolf Reich at the Babraham Institute in Cambridge had defined the first multi-tissue mouse epigenetic aging clock. And we analyzed some samples from a special mouse called the mitochondrial DNA mutator mouse. So this has 2,500 times the level of mitochondrial DNA mutations. It gets a very comprehensive premature aging phenotype. We've shown that with our drugs that basically reduce the level of mutations and reduce these phenotypes. We'd shown they'd reduce the phenotypes, but we wanted to know what was going on with the clock. So that we actually got a surprising results, which was that the mouse didn't show acceleration of the clock at all, despite acceleration of the aging phenotype. So there was a disconnect between the mutations and the phenotype. And the second result was that we did, in fact, slow the clock by 50% in these mice, but it was model specific. It didn't happen in wild type mice. There's an interesting, you know, academic story to that, but that's all I'll say at this point. The result for us was the beginning of a pivot. So, you know, the idea of using the clock was like, let's see if this is the best lever for therapeutic intervention in aging and rejuvenation. But you know, the clock told us, you know, at this time that it's not the best lever. So together with Brendan Swain, we sort of set to work thinking about what's the logical like extrapolation of aging clocks. You know, we're using clocks in a piecemeal way. Like we've got this hypothesis. What does the clock say? There's alternative hypotheses. You know, you can see what the clock says. But for us, the ultimate utility of a clock is to do a systematic screen or CRISPR screen of the genome. So knock out each gene one by one and see what a clock says. And that way you can just basically scan the whole genome and let the genes do the talking, let the data do the talking. And that might reinforce what we know, but it might tell us some really exciting new things. Perhaps there's some new aging biology that outranks the current biology. And we identified a technical solution that needed to be solved. And this was a single cell transcriptomic aging clock. And the reason this was a technical, you know, the problem to be solved was that 10x Genomics had launched a machine called the 10x Genomics Chromium that allowed you to do sort of relatively affordably a single cell CRISPR knockout and get a simultaneous transcriptome. So transcriptome is a very rich signature that you can build clocks in. So, you know, we realized that if we could just find, you know, or create a single cell transcriptomic aging clock, we could do a CRISPR screen on the 10x Genomics Chromium, and we could, you know, basically survey all of biology and see what its link to aging was. And we actually cracked this problem, but we were ready to, you know, basically go ahead and do the CRISPR screen. But then we realized a few things. So the first thing was that we would only be doing single gene knockouts, so we wouldn't have access to combinatorial space. So, you know, when you knock out a single gene, there can be compensation from different isoforms, and it might not be enough to affect a pathway. Really, you want to go in with lots of combinations and see, you know, what affects the biology. The question is, what's linked to aging, not just what's a single gene that's linked to aging. And then the second thing was we looked at our clock, and it was a clock made out of genes, which was, methylation clocks are relatively well talked about, but this was a gene-based clock. And when we looked at the genes, these genes were very interesting because they contained sort of validated aging biology, you know, sort of things that have been linked to lifespan, been linked to aging phenotypes. And we realized this might be a little bit more than just a clock. It might be a window into aging biology itself. So yeah, we applied this clock to an aging time course. We've applied it to other time courses, and now we have a list of candidate genes for rejuvenation, and these aren't pluripotent genes. And I'll just sort of unwrap that a little bit now. So this is really the technical breakthrough and the amount of detail I'm allowed to describe. So what we do is we take a gene expression matrix, so, you know, transcriptome data, And then we do something called dimensionality reduction, which is we basically look at the pathways. So instead of looking at the trees, we look at the whole forest and what's going on. And then what we do is we apply machine learning to the pathways, so sort of our higher level. But the type of machine learning we use allows us to basically have a connection. Well, we're able to basically show the priority of individual genes. So we get a ranked list of genes and their contribution to this machine learning model, which is colloquially or in the academic field called clocks or aging clocks. And when we apply this, to an aging time course, we basically generate a high accuracy clock. So it has a single cell accuracy of 0.94 with a Pearson's correlation coefficient, and you can see we can predict the ages of individual cells. So it's a distribution of age at each time point. This is a publicly available single cell time course for aging human fibroblasts, a bit polarized towards the extremes. But you can see there's individual dots, which are the ages of individual cells. You can see that the older samples seem to have aged, there are two individuals that seem to have aged at slightly different rates. All the cells seem to age, that you don't get these two populations, non-aging and aging. So that's interesting to see. But the more interesting bit is the composition of the clock. So when we look at this clock, we see mitochondrial genes. Mitochondria have got a long-standing relationship with aging. We see ribosomal genes. There's a ribosomal DNA methylation clock. That piece of biology seems to be very linked to aging. We see a gene in our clock that's sufficient to accelerate aging just on its own. Just a single gene. So this was really exciting because we were looking at this and we were like, well, wait a minute, this might be more than just a clock. It might be driving the aging process, or it might be causing at least part of the aging process. And then we realized, well, we've got this methodology, we've applied it to aging just because that's our focus. But why not apply it to rejuvenation, which is not exactly the opposite of aging, but it's it's something we'd really like to have a look at. So you can take publicly available time courses. Shinya Yamanaka has deposited many of these, and you can train a clock, and we call it a rejuvenation clock. So we've got predicted rejuvenation on one axis, and then actual rejuvenation means DNA methylation-based rejuvenation. So that's how we qualify rejuvenation. And we could train a clock with an accuracy of 0.95, so very high. And when we look at the genes that contribute to this clock, we're just very excited because some of these genes are known aging biology. Some of these genes are novel and linked to some quite left field findings that are coming out of the literature. But most importantly, they don't look like pluripotent genes at all. So what we have now is this list of genes which have been given to us by this clock methodology, and we want to throw them into a cell. So basically, just throw them into the cell and see if the genes are as exciting as we think they are. Just to sort of hit home the advantages of using sort of gene-based clocks as a window into aging biology. So firstly, we seem to enrich more causal biology than methylation-based clocks. That doesn't necessarily mean we're a better clock. It's just we seem to have more overlap with what's been defined sort of historically. The great thing about genes is that you can test causality with mature technologies. So you can overexpress a gene to see how involved it is. You can knock it out with CRISPR. These are mature technologies. And the great thing about genes is you can drug them precisely, with the mRNA therapeutic paradigm, or you can target the gene products with small molecules. And so this is where we are now, sort of the next step. We want to weed out the unsafe genes from what we think are safe genes, sort of sitting on our list. So this is sort of where we are at the moment, which is rejuvenating genes, which also have pluripotency. You can play around with the timing and the composition to try and get around it, but the prospect for a greater safety window is really where we want to go and just at least exhaust the possibility of greater safety. So we're quite confident there is a list or a core set of safe rejuvenation factors, but we need to go through this through this experimental process now, and this is actually a subject of a current fundraising. So this is just where we are at the moment with a big red dot. So We've identified targets by developing this gene clock framework. We've protected the framework, which makes it easier to talk about. What we want to do now is basically collide the bioinformatics with the real world, in a dish. Let's see, if this list is fit for purpose. We expect there to be a partial overlap at the beginning. We don't expect the bioinformatics to perfectly represent, what's going on in nature. But we'll be able to get a lot of information from that first collision, see what the overlap is, iterate, change the bioinformatics, see if we get a greater overlap, and then just slowly, reach the best set of factors. And once we do so, that's basically the basis for drug development. So you can start thinking about how are you going to target these genes. Is it going to be mRNAs or small molecules? And then you can sort of decide based on the identity of the genes which indication you're going to pursue. So I've just created this sort of timeline just to highlight a few things. So first of all, once we've got our list of genes or list of safe rejuvenation genes, of course we want to see how we can target them with drugs, but it would also be fantastic to engineer a mouse embryonic stem cell that expresses these safe rejuvenation genes, basically grow that mouse up to adulthood, and then just start expressing these genes and just see what the mouse epigenetic clocks do or the mouse aging biomarkers do. Does it go down? Takes ages to get back up, go down. How long can you keep that mouse going? And then a little bit later, just see, are we moving beyond natural lifespans with this special genetic system? I think these would be really fantastic proof of concept results. And then when we get to phase 1, we actually have the opportunity to sort of measure clocks, aging clocks and human aging clocks in healthy human individuals and see what these therapeutics are doing. So, getting to market approval is a very long road, but getting to phase 1, it's not a short road by any means, but it's a shorter road. It's it's not so far. You could say within this decade. Some of the characters that have been roped into this sort of project. So I've got a background in mitochondrial biology. Brendan Swain, really sort of self-taught on the bioinformatics. He came up with the methodology. We've got all these orthogonal clocks now, so we can triangulate biology from sort of different bioinformatic methods. He's a huge part of this. Romina was a mitochondrial biologist that worked with me previously and came to join us so we could basically take clock measurements in our mice studies. Steve Ives is my dad, but he was a serial entrepreneur, so he brought a lot of business expertise, which meant we didn't make stupid mistakes. And we are actively recruiting right now, so we want to bring in a couple of lab scientists. Basically just to take this list of genes, get it into the dish as soon as possible, and basically get to work, like with the wet lab stuff, and a data scientist, because we'll have these two core activities, basically the lab scientist testing if it works, throwing the ball back to the data scientist, who're going to iterate the methodology and back into the lab. So that's going to be like a big cycle for us over the next few years. Jonathan Milner's supported us from the very beginning. So in Cambridge, UK, he's a fantastic life science angel. He's behind very many interesting companies. Ken Raj has been fantastic. He's just provided us sort of a view of the field and the front lines over a period of time that just means that we're much closer to what we want to do. He's a fantastic guy, and he's a co-author on many of the landmark epigenetic aging clock studies. Wolf Reich allowed us to embed Romina in his lab. He's done some fantastic things besides the multi-tissue mouse epigenetic clock. So he's creating these multi-omic methods which allow you to create aging clocks across molecular layers. So you could go transcriptome, epigenome, many other omic layers I think are in the works, and basically create a list of the best biology, not just the best transcripts or the best methylation sites, the best biology. So that could be a mixture of all of these different things. David's got big pharma experience, and then just some supporters here. So I'm in the Milner Therapeutics Institute, Jonathan's institute, and it's really just a very supportive environment. So you just do the value add bit and they take care of everything else, like taking out the rubbish, just sorting out all of the infrastructure. We recently were accepted onto the Foresight Institute Longevity Accelerator, and that's been fantastic. Provides an open door to, as far as I'm concerned, the best in the aging ecosystem. Creative Destruction Labs is like a Y Combinator, a Canadian version that's decentralized. And I've had some fantastic contacts come out of that. Accelerate Abram's a local accelerator. We've been on a lot of accelerators, and I really think it has helped. And by all means, reach out to me if you're interested in the science or you're interested in supporting us during our next stage where we want to reach these safe rejuvenation genes. We really just want to get to work on these genes now. It'd be such a shame if they were just sitting there on the list, waiting to be revealed. So this is something I'm spending an increasing amount of my time on, but it's absolutely necessary. Thanks everybody for listening.